Whether we like it or not, we're becoming cyborgs. We're becoming transhumans. Transhuman is an evolution from human to post-human, where we're no longer exclusively biological. So the cyborg is having all sorts of add-ons. Are we walking around naked? No, we have clothes. We have eyeglasses. We have earplugs. Now, these are very, very basic, early add-ons to our biology. We're also having much more innumerable add-ons, prosthetic parts, inorganic hearts. We will see much more advanced prosthetics in the years to come. Eventually, we'll have entirely prosthetic bodies.
Human as a design is a wonderful design, but humans as a design have some faults. One fault is we were not designed for the modern age. The industrial age has equipment moving faster than we are, and equipment that's very large that crushes us. So we were designed with an endoskeleton, meaning our bones are on the inside, with our soft tissue on the outside. The world would be better for us now if we had our bones on the outside like an exoskeleton. So we were like a beetle.
Post-human, I think, will be post-biological. By that I mean that it'll be a gradual process. There won't be any sudden transition to a different form. I think we'll gradually integrate more and more technology into our bodies. We'll be replacing our organs with more efficient organs. We'll gradually replace this neural tissue because it dies off after a while. It's easily subjected to chemical damage. So eventually I think we'll replace our brain cells with essentially computerized parts. It'll be much more efficient, much more powerful.
To become cyborgs able to compete with the rapid evolution of computers, we first have to understand how our own brains function. The research in this area has boomed in recent years. One of the factors behind this is the development in scanning techniques. They are now at the level that enables us to observe individual nerve cells at work in living animals.
So is this dying out here? Or
It's in the process of growing in.
The technique is based on fluorescent jellyfish. Dr. Lichtman isolated the gene responsible for the fluorescence. He implanted it in mice and continued his experiments until he had a mouse of which only the brain cells were fluorescent.
They're genetically engineered so that these are now heritable strains of mice. We have a yellow mouse strain, we have a blue mouse strain, we have a mouse strain where only a few cells are labeled, and we're now generating mice that have multiple colors in them. We call them rainbow mice.
With this technique, we can see individual nerve cells make connections with each other and emit signals.
It's really quite jaw-dropping. It's beautiful when you look inside and see these nerve cells that have always been there, but never easy to see before in living animals.
This technique is used now by many laboratories, helping in their efforts to understand how the brain functions. Other scientists use a completely different approach. They take the brain cells apart and let them grow in a small dish on top of a computer chip. The problem with this technique is that it is extremely difficult to place each cell exactly on the right connection point. A team of researchers at IMEC has recently found the trick. They printed a pattern on the chip with a product that the brain cells love to eat. While consuming the product, the cells get stuck to the right spot and their tentacles are guided by the pattern. This way, the whole network of brain cells can be entirely controlled. Some scientists have already been experimenting with such brain dishes. Their chips were less precise, but their results sometimes remarkable.
Let me disconnect the light show.
Dr. DeMars, for instance, tries to communicate with his brain dishes and teach them several tasks.
So each of these dishes contains about 20,000 or so neurons, which are firing away as we speak. So each one's an individual network, and they'll fire spontaneously. We take living rat neurons, and they will rapidly form a neural network. And we have this grid of electrodes underneath the surface of these living neurons, and we can listen to the conversation among the neurons. And we can also stimulate activity within that network. We can send in different patterns of stimulation and look at how the network changes as a result of those stimulations. And that's how we do what we do.
What he does is to teach his brain dishes how to control an airplane.
The network can essentially fly the aircraft in a pretty optimal way. So it won't overcorrect too much, and it'll be able to stabilize it in a wide variety of conditions. I think I've programmed like a 50-knot crosswind into this one. It was what it looks like. And you can see it, the aircraft, when it hits one of these crosswinds, how it begins to oscillate. The oldest dish that I've had is at 2 years. And the only reason why that dish died was because we were moving the lab from Caltech to Georgia Tech. Otherwise, that dish would probably still be alive today. Each dish has a characteristic, so you can learn to recognize one dish from another dish. They're demanding, though. You have to feed them once a week. And
more and more, we will be able to measure and understand how our brain cells function. Deciphering our consciousness is another question. But the experiments of Dr. Yang Dan have proved that in principle, it is possible to read and to understand the brain.
I didn't know that we started this thing again. So this is a digitized movie. We present this to the animal, and we record the activity of their visual neurons. And these electrical signals that we recorded from will travel through these cables.
She managed to tap the vision from the brain of a cat and to reconstruct what the cat sees onto a computer screen.
So this is a digitized movie. This particular movie is a short clip from Indiana Jones, I think.
Here we see the two images next to each other. On the left, the image that was shown to the cat. At the right, the image that is tapped out of the cat's brain.
The picture has a lot of random flickering, the noise. That's probably the noise of the actual neurons, because they sometimes fire spontaneous spikes. We consider those noise, but maybe they reflect something else, maybe the thoughts or something. But we can't distinguish that. So when we use all of those, thinking that they represent visual information, this is the reconstruction that we come up with.
It is the first time an image was tapped from a brain. It proves that we can read and understand the grammar of it. The face is recognizable, but I still can't avoid the impression that the face has something cat-like. Dr. Theodore Berger is head of the Center for Neuroengineering at the University of Southern California. With a team of top scientists from different disciplines, such as neurology and computer programming, they have collaborated to develop a chip that can replace parts of the brain, or, on a longer time frame, add more brain power.
One of the first things that we do in this process is to actually study this part of the brain experimentally. We need to know very precisely how it behaves if we're going to reproduce that function in a computer chip and put it back into the brain. So what we do is to take the brains out of the animals that we use for these experiments and then slice up this brain tissue and keep the slices alive in a dish. And we study the electrical properties of these neurons. Neurons happen to use electrical impulses to communicate with